Now coming to T.H. Morgan's experiment on Drosophila. Drosophila is the fruit fly, those small flies you see around the ripe fruit. Those are the Drosophila flies. So, why T.H. Morgan decided that Drosophila will be the experiment animal or the experiment insect? It is because of the short life cycle. Okay, so only 2 weeks long is the life cycle of Drosophila. The generation time is 2 weeks. Then it grows easily in the lab. Then the males and females are morphologically distinct. Means by seeing we can say that this is male and this is female. The characters that we see can easily say which one is male and which one is female. Then they have visible contrasting traits. Contrasting traits means two uh, different traits of the same character, like here is saying brown body and yellow body. We can easily distinguish between brown color and yellow color. So that is how they have visible contrasting characters, which will make the experiment easier. Then he did dihybrid cross. Dihybrid means two characters are considered in this cross. What are those two contrasting characters? In one cross, he took eye color and body color. Okay. Then in another cross, he took eye color and wing size. Then some of the symbols we must know to understand T.H. Morgan's cross for linkage. So what he did for dominant characters, which is also called wild type. Okay, dominant means wild type. So he used a plus sign to show dominance or the wild type. So here this brown body and yellow body, two colors of the body, two different traits of body color. So here the recessive one is named after this. So yellow color is the recessive one. Yellow color body is the recessive trait, and Y for yellow. So Y, small y is written for yellow body, and Y plus is the dominant one, which is brown body. So symbol for brown body is yellow plus Y plus. For yellow body is Y. Similarly, for white eyes is W, and for red eyes is W plus. It is small w plus. So we can easily see the contrasting characters, and we can easily remember that yellow and white colors are the light colors. So they are the recessive one. While the brown and red color, these two colors are the dark colors. So they are dominant one. This is how we can remember that which one is the recessive character and which one is the dominant character, and the naming is according to the first letter of recessive trait. So Y and W for this recessive trait, and plus is for the dominant ones. Coming to the dihybrid cross made by T.H. Morgan. So he took the female containing yellow body and white eye. So Y and W, and these two chromosomes are the X chromosomes of female, because he took only the characters which are present in X chromosome only. Okay, so this yellow body trait and the white eye trait both are present on X chromosome. He knew that these characters are in the X chromosome. Then took brown bodied male with red eyes. So male is brown body and red eye, and female is yellow body and red eye. And that cross resulted in, see here, same type of gamete will be formed. So this combination will make this one, okay, YW from here and Y plus W plus here, and this chromosome is the Y chromosome male. Okay, so don't get confused. Now this will combine with this Y chromosome and will give rise to this male YW, this Y chromosome. Okay, so this one contains this dominant allele Y plus W plus, which means brown body and red eye. Here there is no dominant, only recessive allele is there, and the one there is no such trait, no such allele present, because it is X chromosome, this Y chromosome. So there is no Y and W allele here. This is yellow body and white eyed male. Now if there is cross between the F1 generation, the female and male of F1 generation, then these 4 types and these 4 types of offsprings are obtained. Okay. Total 8 types of offsprings are there. But according to the law of independent assortment given by Mendel, the ratio wise 9:3:3:1 means 1, 2, 3, 4. 4 different types of individual should be produced. 4 different types of offspring should be produced. But here we are seeing that 8 different types of offsprings are produced. Now let's see what is the reason. See here, we have studied about crossing over. Okay. So if there is crossing over between these two X chromosomes, okay, there will not be any crossing over between X and a Y chromosome. Because they are not homologous. Okay, so there is possibility of crossing over between this X chromosome and this X chromosome of the female. If there is crossing over and part of the chromatid is exchanged, okay, so this will lead to formation of recombinant. Okay, this is Y plus W and Y W plus. And why we are saying this as recombinant? Because this type of combination was not there in case of parent. Okay, here in case of parents we are having YW combination, Y plus W plus combination. Here YW combination, but here we are having Y plus W combination and YW plus combination. Okay, so this type of combination is called recombination because these are not the parental combination. Okay, these YW and Y plus W plus are the parental combination, but here Y is with W plus and W is with Y plus. There is exchange of part of the chromatid, which is called which is because of crossing over during meiosis 1. Okay, and if there is recombination in some of the meiosis, okay, because this is not a compulsory event, crossing over is not a compulsory event. It may occur, it may not occur, and it may occur at any place, not exactly at this place. Okay, so only 1.3% of recombinant offsprings he obtained, and maximum 98.7% were the parental ones. From this he could find that because the recombinants are really few and the parental combination is more, so he concluded that the genes responsible for body color and eye color are closely associated. Because they are closely associated, it is difficult for the crossing over to make recombination. Okay, if there will be crossing over here, okay, in this region. So these two alleles will not be separated, okay. Y and W will not be separated, will not be segregated if there is crossing over at this point, okay. Because they are closely associated, so the recombinant type is few and parental type is 4, okay. Here we can put the genotype. It is YW, it is YW, here Y plus W plus, here YW, here YW, here Y plus W plus. These are all parental combination, YW or Y plus W plus, okay. No YW plus or Y plus W. Here we can find recombinants, okay. It is Y plus W and YW, again YW plus and YW, here Y plus W, here YW plus. So all the 4 chromosomes are recombinant here. Recombinant, cis recombinant, also recombinant, also recombinant, okay. Parental combination is little shuffled here, okay. So linkage is nothing but the close association of genes present in the same chromosome. So these two genes are closely associated, that's why there is less recombinant types we are getting, less percentage of recombinant type we are getting, and more percentage of parental type we are getting, because these two are present closely, okay. If there would have been more distance, okay, if the genes are present at large distances, so what would have happened? Easily there would have been recombination. So the recombinant type would have been like something 10% or 20% or maybe 30%. If the two genes which are present on the same chromosome but are present distantly, okay. So in that case recombinant will be more and parental will be less.